Good afternoon and welcome to the Great Lakes Seminar Series, co-hosted by the Cooperative Institute for Great Lakes Research and the NOAA Great Lakes Environmental Research Laboratory. My name is Mary Ogdahl and I am the Program Manager for SIGLR. It is my sincere pleasure to introduce today's speaker, Dr. Mike Mckay. Mike joined the University of Windsor in 2019, where he serves as the Executive Director of the Great Lakes Institute for Environmental Research and professor in the School of the Environment. Prior to joining the University of Windsor, Mike served on the faculty at Bowling Green State University for over 21 years, expanding his aquatic nutrient cycling research to study the biogeochemical cycling of nutrients, phytoplankton and bacterial community dynamics, and more recently, winter limnology involving research coordination with ice-breaking programs of Canadian and US Coast Guards. Before I turn it over to our speaker, I would like to remind everyone to please leave their microphones muted for the duration of the webinar. You are welcome to enter questions into the chat box at any time during the presentation, and then we will moderate those questions at the end of the presentation. Also, I just want to let everyone know that here in the Ann Arbor area, we're experiencing some really high winds today and some power outages. So if the webinar does happen to spontaneously end, meaning I've lost power, please go back and rejoin the meeting and we will get it restarted. We've got a backup plan in case that happens. So if it does end, just come on back and we will restart. With that, I will turn it over to Dr. Mike Mckay. Welcome, Mike.
Thanks, Mary. Great to be here today. You know, this has been a long time coming, I guess. I was, I think, the first seminar that was canceled or had to be rescheduled after the pandemic started in March of 2020. So again, long time coming. I wish I could say I've got a lot more winter research to share with you since that time, but you know, restrictions in conducting research and sort of moving in some different directions have kept me busy in some other areas. But it's nice to get back into this. And the other thing that's really been I think this is timely. There's a lot of talk right now in the Great Lakes community about winter activities. At the International Joint Commission, my colleagues Michael Twiss and Maggie Xenopoulos, who are part of the Science Priorities Committee, have put forward a working group request to start a working group on winter science. Those of you who attended the SIGLR All Partners meeting a couple of weeks ago will recall that winter biology was an important part of that meeting as well. And there's a new Great Lakes winter science group that's been started that Ted Ozerski from University of Minnesota Duluth has organized, which began actually with support through a SIGLR summit in May of 2019, I believe, which is pushing forward some coordinated winter activities in the basin. So it's a really good time to be involved in winter science right now in the Great Lakes. So I'm going to go ahead and share my presentation. Okay, we can see the presentation all right?
Yes, it looks great, Mike.
Great, thanks. Okay, again, thanks to Mary and thanks Casey Godwin who for his persistence in getting me to present a seminar. Casey told me that there's still a seminar flyer hanging at NOAA from my canceled March 2020 seminar, yeah, and he won't take it down until I actually present one. So here's to changing the flyers at NOAA. And I'd like to also first begin by acknowledging the various sponsors, supporters of this research. This involves a number of scientific collaborators, the Melee Research Group. Some of you may be on the line today. That core group began in 2007 with Steve Wilhelm and George Beauregard and Michael Twiss and Hunter Carrick. And a number of others, and has grown since that time. Collaboration between ice-breaking platforms of both the U.S. and Canadian Coast Guards. So we've done work extensively with the Griffin on the Canadian side and with Nia Bay on the U.S. side, and a number of agencies, organizations for their financial support of this work. And I'm showing their logos on this page. All right, so again, most of us working in the Great Lakes Basin, this is a typical scene from our summer research. This is actually an image from Put-in Bay in the Lake Erie Islands of the Western Basin. And unfortunately, this is a scene that a lot of us are far too familiar with as well. Again, phenomena that occur in the summer and the fall, these being in this case the expansive or harmful algal blooms we find that recur annually in the Western Basin. If you look at pretty much any parameter, sorry, I'm just trying to move the toolbar here so I can see my own slides. Apologies. We can think of almost any parameter, and we will have some data on that parameter typically between the months of April and November or so. Absent from most of our understanding of the Great Lakes and many aquatic systems in general is what happens in these shoulder seasons and basically the winter in these north temperate lakes. So today's presentation, a little table of contents here, talk a little bit about ice cover in the Great Lakes, a brief history of winter work on Lake Erie, then segue into some of the field surveys we've conducted since 2007. So we've been at this for over a decade now. Going to share with you a couple of stories from that research and then finish up with some plans for the future. All right, so ice cover in the Great Lakes. This is showing you average ice cover in this image over the period 1973 to 2002. And as you can see, despite Lake Erie being the southernmost of the Great Lakes, it's the lake that typically has the most ice cover. You can see here, scale-wise, probably maximum ice cover 80, 90% or more, typically on Lake Erie. And that's mainly because of the shallow bathymetry of the lake. Ice cover is declining in the Great Lakes, so paralleling what we are seeing in the Arctic, and this actually is a figure adapted from a manuscript by Jia Wang from Noah Glurl, describing reductions in total ice cover in the Great Lakes of about 70 to 75% from the period early 1970s through to present. As you can see, the ice cover reduction is not linear. Ice cover is highly variable within the Great Lakes, but likely we're seeing an increased frequency of lower ice shears, thus leading to this overall reduction in total ice cover. A little history of winter research on the Great Lakes. When we were first starting into this work, we conducted a literature review to see what was known, and came across a series of papers by David Chandler. He was, I believe, perhaps the first director of Stone Lab, Ohio State University's freshwater lab in Lake Erie. And you can see here, even from the early 1940s, well, probably late 1930s, there's a lot of school spirit at Ohio State. You see the Buckeye on the ice shanty off the coast of Stone Lab. And so conducting both winter and summer research in Lake Erie off of Stone Lab, you can see in the wintertime with the ice shanty, probably in addition to ice fishing, also sampling the lake beneath the ice. And there are a series of papers that were published in the Ohio Journal of Science providing data on phytoplankton, zooplankton, nutrients of Lake Erie throughout all seasons. Moving on now to the mid-1980s or so, and this actually was work that was conducted by scientists at Noah Glurl, and I would imagine Hank Vanderploeg may be on joining us today. He was one of the participants in this research. So work on Grand Traverse Bay, setting up a winter field station at this location. And I believe the motivation behind that work was to look at factors involved in the success, the overwintering success of whitefish that is spawning in the fall and overwintering under the lakes. And so that provided a rationale for this work, but again provided a lot of interesting information on what life is like under ice in the Great Lakes. At the federal level, the US EPA determined that we should be looking at the winter season, and they actually moved to helicopter surveys over several winters in the 1980s. That program was stopped, however, due to safety reasons, with a requirement that they moved to dual engine helicopters, and they only had single engine helicopters available. This actually was some motivation for us doing work. This was from 2015, I believe, March 2015. You recall 2014, 2015, very robust, severe winters in the Great Lakes system. We had roughly 20 inches of ice at this location in the central basin, about 10 nautical miles north of Cleveland. We had a helicopter land on the ice and drop myself and my grad student off to spend several hours taking course through the ice and sampling in Lake Erie. So that brings us to the present work conducted by my colleagues as part of the Melee network initially. And this was conducted through Operation Coal Shovel. So Operation Coal Shovel is a joint operation between the US and Canadian Coast Guards in support of keeping the Great Lakes open for shipping during the winter. So unlike the upper lakes, basically Lake Michigan, Lake Huron, Lake Erie are open for business during the winter. Commerce continues. We've got Lakers moving between ports, moving cargo between ports, and it's the joint effort of the Coast Guards to maintain ice-free passage for these for commercial transport. So I mentioned earlier, we've been working with the Coast Guard ship Griffin on the Canadian side. And this is a picture taken from the bridge of the Griffin, a relatively large, light ice-breaking vessel with reinforced bow, capable of breaking through ice in most of the Great Lakes. And on the US side, most of the work has been conducted in coordination with the Coast Guard cutter Neah Bay, whose home port is in Cleveland, Ohio. And this is one of about 10 of these ice-breaking tugs that are used extensively by the US Coast Guard, again, as part of Operation Coal Shovel in the lower lakes. So we started a collaboration with, or partnership with the Coast Guard and the commanding officer and crew of Neah Bay in 2009. And this is a picture showing, at the time, commanding officer Bill Waitira. Bill's actually now the commanding officer of the Coast Guard cutter Polar Star, which is the only heavy icebreaker in the U.S. fleet, regularly making runs to Antarctica out of its home port in Seattle. But during the time from 2009 through 2011, Bill was commanding officer of Neah Bay. Bill also had a science background. He had a master's of oceanography from the University of Rhode Island, was interested in conducting scientific research, and so this provided a great outlet for him to satisfy some of his curiosity working while he was stationed on the Great Lakes. What we did in this case, because the Coast Guard is a military unit, part of home security in the U.S., it's not always easy for us to join surveys of the lakes, and so we basically provided Bill and his crew with equipment for both sampling and processing samples, and they turned the ship's office into a mini laboratory. So you can see here he has one of the crew members who is filtering water samples for chlorophyll, biomass, and nutrients during opportunities of sampling in the Great Lakes. This picture shows Bill during one of the ice safety exercises Neah Bay conducts every year. During this exercise, he has probably his executive officer and a number of crew members off performing ice training or ice rescues, likely throwing a crew member into the open waters of the lake and practicing rescuing. While they're doing that, Bill took the opportunity of boring holes through the ice and measuring light penetration through the ice for our own research. And through the partnership with the Coast Guard cutter Neah Bay, we have been able to increase the temporal frequency of sampling during the winter. So this is a paper we published in EOS in 2012 showing bearings of Neah Bay during the winter, the blue dots, and you can see Neah Bay, again, home ported in Cleveland, spending a lot of time making the transit between Cleveland and the mouth of the Detroit River, usually escorting ships during those transits, and then spending time in the rivers. In addition to breaking ice in support of commerce, Neah Bay and his fellow ice-breaking tugs are also involved in breaking ice in support of reducing coastal flooding. And so spending a fair amount of time in the St. Clair River, Detroit River to reduce flooding impacts. And then the red dots are showing actual locations where Neah Bay sampled for us. I believe this was from 2011. A lot of challenges to working in the winter, as you can imagine. Equipment freezing up. On the left side, you see my colleague Michael Twiss of Clarkson University. Michael's deploying a plankton net off the side of Griffin. And as you bring the net back up out of water, starting to freeze. On the right-hand side, you can see a couple of the crew members who actually, one's a tech op for Environment Canada, the other one a crew member of Griffin, working with a Niskin bottle. And again, problems you have with Niskins with mechanisms starting to freeze to pull them out of water. And just ice jamming up against the ship. So it's really a collaborative effort in sampling during these surveys. In this case, using poles to move ice away as we're deploying equipment. We've had the opportunity to deploy sediment traps in Lake Erie under the ice. We did this for a couple of years in 2010, 2011. These were just passive sediment traps, essentially acrylic tubes that will passively collect material that's exported through the water column during the winter. And here's where we pulled one of them up and trying to figure out how to detach it. So these early surveys conducted in Lake Erie in association with both the US and Canadian Coast Guards showed us there are expansive blooms of psychrophilic algae within the lake. So really, one of the first things we saw in 2007, breaking ice, mainly in the central basin of the lake, was this brown water gushing up as we're breaking through the ice. And lo and behold, this wasn't sediment. These were mats of diatom algae, centric filamentous diatom algae, as you can see here. And these are mainly of the species Alocasyra islandica, as well as Stephanodiscus binderanus. And these are two scanning electron microscope images of these diatoms. So centric diatoms, filamentous diatoms, very common during the winter, also during the spring, persisting into the spring in Lake Erie. And our initial surveys showed us that there's biomass in the ice, under the ice, and in open water of Lake Erie during the winter, and showing you a list of some of the publications that have come from this research since our earliest studies. In a summary of those studies, we reported that there are, again, expansive blooms of winter phytoplankton, mainly diatoms. These diatoms are nutrient sufficient based on nutrient stoichiometry of the cestin, also based on ectoenzyme activity assays showing that they are usually phosphorus sufficient. Low light adapted, you'll see that coming later in the presentation, generally physiologically robust. The fate of this production, some of you would have joined us during the Sigler supported hypoxia summit earlier in the fall. And there is discussion during that summit about the role of these winter-spring diatoms in the formation of hypoxia in Lake Erie's central basin. So on the right-hand side, you see this image showing you zones of hypoxia in the central basin, partly a function of the morphometry of Lake Erie, where the central basin is isolated, or the hypolimnion from the central basin is isolated from both the eastern and western basins of the lake, relatively thin hypolimnion, diatom carbon exported during the winter into the spring. That carbon accumulates in the hypolimnion of the central basin and is remineralized, and in doing so, oxygen is being exhausted and not being replenished. And so the Wilhelm et al. Article from 2014 describes some of the sediment trap work that we did, and also some bacterial production work associated with that, showing that rates of bacterial production during summer are several orders of magnitude higher than they are during the winter. So there's a temporal staggering between the production of carbon that's eventually remineralized that results in hypoxia during the summer. Also work done by Ewan Reevey, Michael Twiss and colleagues in this space as well. All right, so moving on now to a couple of the, I think, interesting stories that have developed based on our winter research. And the first one comes from a paper we published in the ISME journal in 2013, Winter Diatoms as Ecosystem Engineers Promoting Ice Formation as Ecological Refuge. And I'm going to come back to this graphic later on, so we'll move on. And this was mainly the work of my doctoral student at the time, Nigel D'Souza. Nigel is now an assistant professor at the University of Louisiana, Lafayette. Okay, so this came from some observations we made during our early winter research. We were hoping to be able to take some of the winter diatoms that we had found in the lake in the Central Basin, bring those back to our lab at Bowling Green, and do some culture work so we could really characterize the physiological features of these psychrophilic diatoms. To do so, we decided we would make media based on Lake Erie water. So we had filtered lake water that we processed on the cruise. And because these Coast Guard boats are not scientific research vessels, they're working boats, they don't have a lot of the amenities that a research vessel might have. And so, there are no walk-in fridges or freezers for our use. But since it's winter, we decided we'd just store things in coolers on the deck. So we have filtered lake water. We collected again to make media back in the laboratory. At the end of the cruise, pulling the filtered lake water, packing it back into our vans, we find that it's still liquid. We also have net plankton. So cestin we collected by plankton net, suspending that in lake water and using that, we would be using that as inoculum to start cultures back in the laboratory. These samples with net plankton were often frozen solid. So Nigel was at an IAGLR meeting in Toledo, I believe it was 2009, and he ended up speaking with Rick Lee, a professor at the time at Miami University in Oxford, Ohio. And Rick is actually an expert in the area of biological ice nucleation. And so Nigel's explaining some of his winter research to Rick and discussing these observations he's making about these plankton freezing, consistently freezing on the deck of the ship. And Rick, thinking about this, thought that, well, the discrepancy, reconciling this discrepancy, this really sounds like a case of biological ice nucleation. And you may be familiar with biological ice nucleation in nature, relevance to cloud seeding, frost damage to plants. Artificial snowmaking often relies on lyophilized bacteria that are known biological ice nucleators. This graphic on the right actually is a graphical abstract from a paper we published in ES&T in 2018, looking at the presence of ice nuclei actually coming from terrestrial ecosystems. This actually is the Maumee River watershed. We can measure ice nuclei being aerosolized from a weir at Grand Rapids, Ohio. The method we used for this, collecting plankton using a net, and then running ice nucleation assays, and the assays come in different ways. A freeze drop assay, basically perspex block that we can progressively lower the temperature on. There's a CCD, charge-cooled device camera over top, taking photos every 30 seconds or so, and actually visually identifying samples freezing and correlating that with the temperature which they froze. Another method, this is a method that we actually use mostly working with Rick Lee at Miami University, a microcapillary method, filling microcapillary tubes to which we have a thermocouple attached, and then measuring the latent heat of crystallization. So as something freezes, it releases heat, and we can measure that latent heat of crystallization to determine the freezing temperature. And then controls for this would include filtered lake water, purified water, so molecular water from the laboratory, and then also as a positive control, ice nucleation protein from one of the classic ice nucleating bacteria, Pseudomonas syringae. Okay, so just focus on the left here. We're looking at temperature of crystallization for Great Lakes phytoplankton, and these are samples from Lake Erie from April 2010, from February 2011, April 2011. These here you're seeing other lakes, Lake Superior, Lake Ontario. First of all, look at the light gray boxes, and this is purified lab water and molecular water. And we're finding the temperature of crystallization close to -20 for that control. And then when we take filtered Lake Erie water, freezing at slightly warmer temperatures around -15. And then we have samples containing cestin from Lake Erie. These would be the filamentous diatoms from the lake. And we're seeing that the temperature of crystallization ranging from around -10, -12, all the way up to -2 or -3, which actually is the classic temperature of crystallization for an ice nucleating bacteria such as Pseudomonas syringae. Nigel then took samples from the lake and used a modified scanning electron microscopy protocol. So typically when you do scanning electron microscopy, you remove all organic material using an acid cleaning. This protocol was slightly more gentle, leaving some organic material on the samples. And it's not, this is not too surprising that you would have bacteria associated with the surface of these diatoms. There's many known examples of diatoms and bacteria living in concert. The identity of these bacteria, we use both classic microbiological methods as well as 16S sequencing to determine that these bacteria were indeed members of the Pseudomonas. So various species of Pseudomonas attached to these diatoms. When we take pure cultures of these bacteria, both in 2010, 2011, culture the bacteria and then test them separately for their temperature crystallization, we find that some of the bacteria are both ice nucleation positive and ice nucleation negative, with their temperatures of crystallization coming in as you would expect for Pseudomonas syringae, maybe sometimes a little bit cooler, but typically in that range, about -3 degrees Celsius. And again, our controls down here with the molecular water and the filtered lake water, molecular water and medium. So ecological implications of ice nucleation with diatoms. So think of diatoms. Diatoms are non-motile photoautotrophs. They need to be suspended. So during the period of ice off, diatoms are typically suspended within the mixed layer. Because they're in the mixed layer, they're never too far away from the surface, and so they typically obtain enough light. But it's much like a pot of vegetable soup. If you don't stir things around, everything's going to sediment to the bottom. Same thing with diatoms. So typically suspended by turbulence. But what about in a non-turbulent or relatively non-turbulent or ice-covered environment? Well, there was work done on Lake Baikal to demonstrate that convective mixing could be implicated here in maintaining diatoms relatively close to the ice cover. mixing anywhere from 2-3 meters, maybe 5 meters in depth. Another possibility is that there's actual physical attachment to the ice cover. So I'm going to show you two models. One is a model that was first proposed by Dave Garrison and colleagues in 1983, published in Nature. Some of you may know Dave Garrison. He was a longtime program director in biological oceanography at NSF. And he proposed this physical mechanism by which diatoms can be partitioned to the underside of ice in the coastal ice shelf off of Antarctica, for example. So if you take ice cores off of Antarctica or the Arctic ice shelf, you'll typically find a colored zone at the bottom of the ice core. The ice is porous and actually provides habitat that's colonized by diatoms and other organisms, and the coloration is typically because of diatoms that are living within that ice. And so he proposed a mechanism by which diatoms can colonize the underside of ice, and this involved supercooling of the water column, fragile ice forming. Fragile ice is very sticky, sticking together, sometimes forming anchor ice, which can scour the sediment, attaching to diatoms that may actually be sitting on the surface of the sediment, eventually sloughing off of the sediment and rising to the top and attaching to the undersurface of ice. So we took Garrison's physical model, and we simply added a biological element to it, so biological ice nucleation. We know there's supercooling occurs in the Great Lakes. There's a number of papers that have discussed supercooling occurring, especially in shallower waters of the Great Lakes. So mechanisms by which fragile ice may be formed. We also now know, based on this work, that diatoms and their associated bacterial partners can also induce ice nucleation. So between fragile ice and the diatoms, we can get ice formation. Again, ice sticking together. We can get anchor ice formation, scouring of the sediment. We know there's lots of these diatoms. They're meroplanktonic. They spend a portion of their life cycle on the surface of the sediment. So scouring the sediment, eventually sloughing off, rising, and attaching to the underside of ice. And so a mechanism by which there could be actual physical attachment of the diatoms to the underside of ice in Lake Erie. And we were able to show this, demonstrate this in a laboratory experiment. I have a video here I'm going to try to play. So I'll set this up though. We have two water jacketed beakers here, one of which contains filtered lake water on the left. The other one contains filtered lake water to which we've added some of the diatom cestin that we've collected from the lake, and then progressively lowering the temperature in each beaker. And we'll see if this video will play. Okay, so presumably you can see the video. It's a time lapse. The whole video takes about 15 minutes or a little bit more. So we're looking in both cases, we still have liquid water, but we're progressively lowering the temperature here. And in the right-hand beaker, you should start seeing some slush being formed. Right there, you can see the slush being formed, but not in the beaker on the left, which contains only filtered lake water. And when we collect that slush, we find there's actually partitioning of the diatom, the filamentous diatoms into that slush. Okay, I'll stop this. And go back to my presentation. Okay, we're back on the slide, all right? Okay. Okay, so that's the first story I wanted to share with you from that resulted from our winter research in Lake Erie. The second one it was resulted from work that was conducted in 2010, 2011, and then 2012, a very mild winter in the lake. So ice cover is a factor driving microbial community composition in Lake Erie. And this work was published in 2016 in the journal Environmental Microbiology, and actually got the cover photo on the journal of that issue. And leading this work was my postdoctoral fellow at the time, Ben Bell. Ben is now an associate partner with a environmental consulting group in Vancouver. So what you're seeing here is a MODIS image of Lake Erie from February 15th, 2012. As I mentioned, a very mild winter within the Great Lakes Basin, and ice cover was negligible during that winter. And so lakes and reservoirs can serve as rapid responding sentinels of human influence on the natural environment, rendering them powerful tools to advance our understanding of a changing climate on ecosystem structure and function. And the Laurentian Great Lakes are particularly valuable in this respect in that they share characteristics of both oceans and closed basin systems, such that knowledge gained from their study can be used to gain insights for our coastal oceans. So this study, as I mentioned, we used surveys from 2010 to 2011. These were both sort of, I would call them regular ice years in Lake Erie. What you're looking at here is total accumulated ice coverage, or TAC, for Lake Erie from 1973 through, in this case, I think 2015. The green line is showing you median TAC ice coverage from that period. Now, these percents on the left here don't look very high. That's because TAC actually takes the ice cover for the system over an aggregated period of time, in this case between December 4th and April 16th. And so typically, again, during the height of the ice season in Lake Erie, we'd see 90–95% ice cover. But when you look at it over the period from December through April, that ice cover is lower. And so again, 2010, 2011, relatively sort of regular, normal ice cover years in Lake Erie. Working with both the Coast Guard ship Griffin and the Coast Guard cutter Nia Bay, we got both high spatial and temporal coverage. So the Griffin, we would often get a week in mid-February, being able to cover western, central, and eastern basins of the lake. And then working with Nia Bay, higher temporal resolution, with sampling usually occurring between December and into March or April in the lake, but typically not as much spatial resolution sampling. And then 2012, I mentioned a very mild winter within the Great Lakes system, and you can see here very low ice cover through Lake Erie at that time. And this again taken middle of February in Lake Erie, roughly similar coverage in the lake, both between working with Griffin in February 2012 and our work with Nia Bay again between December 2011 and March or April 2012. Okay, looking at physical chemical properties between the sort of conventional ice years, normal ice years, and the low ice year of 2012. Temperature between the 2 years through the water column doesn't vary too much. Slightly warmer in 2012 compared to 2010, 2011. Isothermal in both instances, as you would expect. Dissolved oxygen, again, really not much difference between the two, fully oxic water columns. The big difference comes from turbidity. When we look at the ice-covered system, 2010-2011, we have a high transparency in the water column, turbidity of less than 2 NTUs. 2012, no ice cover, very turbid system, 14 NTUs measured at central basin stations during that winter. Looking at nutrients during this time, nutrients were also elevated. So what we've done here is we've actually exploited a program from the province of Ontario. The Municipal Water Intakes Monitoring Program has been active since the late 1960s, using samples from municipal water intakes in all of the Great Lakes that border Ontario. And those samples are analyzed for nutrients and also to some degree phytoplankton at roughly 2-week intervals throughout the year. So we've got intakes here that represent the 3 basins essentially, while Lake Erie inflow from Amherstburg. The Elgin water plant, the intake extends out about 1 kilometer into the central basin from that site. And then at the eastern end, leading into the Niagara River. And what you're looking at here is SRP, soluble reactive phosphorus, as well as silicic acid from these various locations. The shaded area is a long-term range over about 10 or 15 years, I believe, for these locations. And then the solid lines represent what the nutrient dynamics were in 2010 and 2011. Normal ice cover in Lake Erie during those times. 2012, again, being the negligible ice cover in Lake Erie. And what you see here is elevated SRP in 2012 compared to the other years. Elevated silicic acid in 2012. Declining, mainly presumably because of consumption by diatoms during the winter. But this idea that, and I'll show a slide a little bit later, without ice cover, there's little chance for nutrients, especially particulate nutrients, to be consolidated in the sediments. And wind-aided turbulence continues to resuspend material from the lake, likely maintaining elevated nutrient concentrations during warmer winters. Additional implications of ice decline, we saw lower phytoplankton chlorophyll biomass. So this graph comes from data provided through the US EPA through their April surveys, and these are data available through the Glenda website. 2012, low ice winter. We see this low chlorophyll A biomass and these are central basin stations of the lake. Compared to 2011, a more normal ice cover. Go back to a previous low ice winter, 2002, we see the same trend. Low chlorophyll biomass in 2002, either side years of higher ice cover, and we see the biomass increasing. This was interesting. So we would typically look at size fractionated chlorophyll biomass as part of these surveys. We would use 0.2 micron and 20 micron pore size filters. 2010-2011 surveys, we find that most of the biomass is in the 20 micron fraction, and you can see that here. So the black squares, we find unity here between the two axes, and so a strong microphytoplankton contribution. That makes sense because these diatoms are often filamentous and oftentimes form mats almost, and would be collected, be trapped on the 20 micron filters. So most of the biomass during the winter collected in 2010, 2011, normal ice cover years would be large biomass. 2012, we see a deviation from this trend, moving towards smaller cell sizes. So we saw a large decline in the biomass of the filamentous chain-forming diatoms of Olicosyra islandicus, Stephanodiscus binderanis, and roughly an 80% decline in the biomass of those diatoms in this warmer winter of 2012. We believe the phytoplankton biomass may have been restricted in 2012 because the phytoplankton, particularly the large diatoms, were likely light limited during that winter. And what you're seeing here are metrics of photosynthesis. So we would run photosynthesis irradiance curves using C14 uptake. You can see here, IK is the light saturation level for maximum photosynthesis, relatively low levels. In other words, these are low light adapted samples from both 2010, 2011. These are all the same site in the central basin of Lake Erie. In 2012, a few things jump out. One is this extinction coefficient for light, much higher, an order of magnitude higher than we would see during years where there's ice cover in the lake. Again, owing to the turbulence that we see, a lot of sediment resuspended in the lake. And the other one is the light penetration through the mixing zone. What we see here is that light level is lower than the IK value, meaning that the cells cannot attain the enough light to actually grow at their maximal rates. So, evidence suggesting that there was light limitation occurring during this mild winter of 2012. So conclusions for this section on a mild winter. We're seeing an increased frequency of low ice years in the Great Lakes. Implications of low ice cover, increased turbulence, elevated nutrients, a reduced light climate. We're seeing a shift in phytoplankton community size structure, moving from these filamentous diatoms when we see ice cover to an increase in small-sized algae during the milder winter. We saw a higher abundance of cryptomonads, for example, and the same was found during the subsequent EPA spring survey in April of 2012. And this may have food web implications. That was one thing we were not able to study as part of this project, but it would be interesting to see what the cascading food web implications of that change in community size structure would be. And just briefly, this idea of a new normal, this actually is from February 2020, so not, the not too distant past. And again, a year of a mild winter and very low ice cover in Lake Erie. So implications of declining ice cover, as I mentioned earlier, we may be seeing an increase in the frequency of low ice winters in the Great Lakes, at least over the past 20 years or so. And there are a number of implications for the declining ice cover in the Great Lakes, and you can see here this infographic from the Cleveland Plain Dealer from a few years ago. Everything from reduced erosion in coastal areas because of ice cover to protection of habitat, or for overwintering eggs of fall spawning fish such as whitefish. Implications for evaporation in the Great Lakes, implications for snowfall. You can see here the erosion. We've got some examples actually close by Windsor. This is located close to Wheatley, an old coastal road that's being eroded partly because of the lack of ice cover we've seen in Lake Erie in recent years. This is a paper actually from Don Scavia from 2019, where they were suggesting that Lake Huron can be a source of phosphorus for Western Lake Erie. And they postulate that during mild winters, and you can see here a MODIS image from 2012, again, that mild winter I focused on for Lake Erie, same thing for Lake Huron, we may start seeing sediment plumes occurring because of erosion. And we have a water intake located at Sarnia, Ontario, that they pulled data from to show that there was elevated phosphorus during this low ice winter compared to previous winters with higher ice. Same thing was done by Ken Nichols in a paper published in LNO in 1998, again showing a correlation between elevated nutrient concentrations. These are again data taken from Point Edwards, Sarnia, the water intake, elevated nutrient concentrations during low ice years. So next steps for us, we have been doing some metatranscriptomic analysis of the diatom blooms in Lake Erie. We currently have a project with Steve Wilhelm, University of Tennessee, where we have 90 metatranscriptomes being developed through the Department of Energy's Joint Genome Institute. Most of those have been returned in the past month or two, and so we're working on with Steve on reading those transcriptomes and interpreting them. Another thing I wanted to mention was this upcoming Great Lakes Winter Grab. I mentioned it earlier. So this is being sponsored in part by one of these Sigler Summit series and led by Ted Ozerski from University of Minnesota Duluth. And this will be happening in February 2022, and we are still soliciting participants. So if this is something of interest, please feel free to get in touch with me, and I'll pass your information on to Ted, or reach out to Ted directly. We have a group that's been meeting informally, roughly monthly, over the past 5, 6, 7 months, working to planning this grab, and we're trying to have representative sites sampled throughout the Great Lakes Basin during a period of about 1 week in middle of February of this coming year. The metatranscriptomes, I just want to mention, we did publish a paper in 2016 in JGLR based on some metatranscriptomes that we had made following sampling in 2010 or 2011 during a typical ice-covered winter. A few traits, that we identified from those metatranscriptomes. There's a high number of reads associated with light harvesting proteins. You can imagine organisms living in a environment of lower light, putting more emphasis on producing light harvesting proteins. We also found there was some upregulation of genes involved in cold adaptations such as desaturases, which would be involved in increasing membrane fluidity. Interestingly, we found no evidence of any genes or upregulation of genes involved in ice-active proteins. A number of marine diatoms that we find from coastal Arctic or Antarctic communities often possess ice nucleation genes. We did not find any evidence of these, despite the fact that we've demonstrated strong ice nucleation activity associated with the diatoms of Lake Erie's central basin. Part of the reason being is that there's a lot of diversity in ice nucleation genes, and we may be looking at something that's not actually even a gene product associated with the ice nucleation activity there. And we've also become very interested in looking at sort of unconventional mechanisms associated with the decline and fate of ice-associated blooms. So this initial transcriptome analysis that we published in 2016, we found evidence of some pathogens, including oomycetes, chytrid fungi, as well as viruses that could be important in the decline and fate of the winter bloom in Lake Erie. And so working now with various colleagues, including a postdoc, Caitlin McKinless, who's actually physically working with Tim James at the University of Michigan on chytrid infections of these diatom algae. We have diatoms and their associated chytrids isolated from Lake Erie's central basin from both 2019 and 2020. And Caitlin is conducting investigations looking at factors influencing the rate of chytrid infections on these diatoms. And with that, running out of time, I'm going to stop it and be happy to answer any questions you may have. Thank you.
Thank you so much, Mike. That was a really interesting talk. So we've got time for a few questions, and we have some entered in the chat box. So we'll start there and see how much more time we have. First question is from Casey Godwin. He thanks you for this interesting seminar and asks, as part of the culture work with diatoms and their associated bacteria, did you find any evidence for fitness consequences associated with forming ice?
Yeah, so the one thing we never really got, so it's been very difficult to establish cultures. Now, when the early work was done with Nigel, we were mainly trying to establish cultures, growing them at 4 degrees Celsius essentially. We're able to maintain cultures, but typically only for a month or two. The more recent work with Caitlin, where we've actually, again, these are diatoms that we collected from the Central Basin during the winter, have cultured. These are probably better described as maybe psychrophilic algae, in that they're actually tolerant to a wider range of temperatures. And so she's been growing them at 4 or 5 degrees Celsius, but also at 13, 14 degrees Celsius. So we haven't done exhaustive studies to look at fitness associated with them, in part because of, early work with them suggested that they were very fastidious. And so hopefully moving forward, those are some things that can be addressed.
Great, thank you, Mike. Next question is from Bopi Badanda. He says, in the Antarctic and to a lesser extent in the Arctic, the under ice algae support a vibrant food web in the winter that flows up the food chain, for example, from krill to whales. Do you suppose similar connectivity exists between ice algae and food webs in the Great Lakes? And what is your take on the ice-lake life relations, as ice habitat in the Great Lakes is likely to decrease in the coming decades?
No, thanks, Bopi, and thanks for joining today. Yeah, I mean, the structure of ice is quite different, right, between marine settings and lake ice. Ice we find in coastal marine zones is porous. There are microchannels, and it provides habitat and maybe helps to support more of a vibrant system associated with the ice. I think they even have a name for it, the symphagic habitat. Lake ice, again, doesn't really have much structure to it. But again, the ice nucleation activity associated with these diatoms suggests that there may be some ways it can attach to the bottom of the ice. Now, we have, going in with an icebreaker is sort of like a bull in a china shop. So we don't have a lot of good information about structure of the ice and the communities associated with them. We've done some work where we've augured holes through the ice and taken samples, but that's not really been so intuitive in looking at structure of communities. The question timing is good. There was actually a paper out in PNAS in the past week or two from a group at McGill and at the University of Quebec at Chicoutimi, looking at the under ice ecosystem, focused mainly on the zooplankton community. In that paper, they do suggest that there can be a fairly vibrant overwintering community of consumers. We certainly would identify zooplankton, a lot of copepods, when we pulled up plankton nets, and certainly see evidence of diatoms within the guts of copepods. But again, that's something, these community interactions really need to be fleshed out, and that's something that we'd love to see more people putting effort into.
Thank you, Mike. We're at 1 minute till 3 o'clock. I think that we will take one more question. And after that, we've got several others that I don't think we're going to have time to get to, but I will download those questions and send them to Mike, and he can follow up with you, if that's okay.
Thanks.
So our final question will be from Bob Heath. He asks, could the ice-forming proteins be produced by the admate bacteria instead of from the diatoms?
Thanks, Bob. And again, nice to hear from you. Bob was the professor at Kent State when I was first starting at Bowling Green, and got to meet him early in my career, and he was always very supportive of the work. And I think he'd always make comments about the cool things we got to work on. So nice to hear from you. Thanks for the question. Certainly, the fact we have these, I think they're probably mutualistic bacteria associated with the diatoms, would suggest, and knowing that they're Pseudomonas, would suggest that the ice nucleation activity would be attributed to these partners. We were just never able to identify the genes associated with the Pseudomonas partners. And again, there are these classic INA genes associated with terrestrial Pseudomonas syringae. It's very possible there are other ice nucleation genes that just weren't available in the databases when we were doing this work. So I strongly think it probably is attributed to the bacterial partners. In this case, we just weren't able to identify the genes.
All right, thank you very much, Mike. Thank you again for spending the time with us today and talking to us about your work. And thank you for attending, everyone that is here. And please keep an eye out for our upcoming seminars. We have approximately one a month coming up. I believe that our schedule is up on the Sigler website. And if you are not receiving the seminar announcements via email, please reach out via our website and we will get you added to the list. Thank you, everybody. Have a great week, and thank you again to our presenter. Take care.